This video introduces some of the basic concepts involved in descriptive statistics. Life is variable. No two things are exactly alike. When you want to compare data, for example, height in males versus females, how do you know if they are really the same or different? Because of variability, if you just picked an individual from each group, you might pick by chance a short male and a tall woman. You might conclude from this that women are taller than men. Maybe we need more people, a larger sample of the population. If we measured a lot of people's heights, we would see there is continuous variation in height. We are not built to just be one height. But also, the data would vary because there is variation within an individual based on the time of day the person was measured, early in the day versus late, or our ability to measure it, the accuracy of the measurement device, and simple measurement error. So, how do we know who is taller? Because data are variable, and usually we can't measure an entire population, we use statistics to help us describe and compare data. You will need these tools specifically for your projects in this class, but also for doing your own research and interpreting other people's data if you stay in science. If you don't stay in science, understanding statistics can help you interpret things you hear in the news, such as political polls, results of clinical trials, betting odds in sports, and so on. Often we can't measure a whole population, such as the entire population of humans on the Earth, or even in the United States, or even at Smith. Thus, we use a sample and knowledge of probability to make predictions about entire populations. We estimate values and then make predictions as to how good those estimates actually are. So if we want to test if men are taller than women, but couldn't actually measure the height of every person in the whole world to find out, we would take a representative sample. Sampling is important and used all the time in political polls, censuses, lots of different kinds of research. It's very heavily used in biological research. Part of the key here, too, is to figure out how to get a representative sample so that you're not biased in the sample that you get. So let's say we do take a sample and we want to describe our sample. We need some way to do that. So descriptive statistics are what we use to describe our sample. One thing we need to do is to say something about what the typical individual is. That's our value of central tendency. It gives you a sense of what the typical individual is. There are three very common used measures of central tendency. One is the mean, which is the average value, which adds all the values together and then divides by the sample number. The mode is the value that most individuals have. It would be the peak of your distribution. The median is the value where there is an equal number of individuals above and below. It's the midpoint of your sample. The mean, median, and mode can all be the same value if the distribution is symmetrical. So here's a symmetrical distribution. It's a normal shaped or bell-shaped curve, and you can see that all our three values are exactly the same because it's symmetrical. So your peak is the same point as your average, which is the same point as your midpoint. When the data is skewed and not symmetrical, like your positive skew or negative skew, you can see that the values are not the same. So in the case of positive skew, our mean is greater than our median, which is greater than our mode. This is important to note because if the distribution is symmetrical, it doesn't matter which measure of central tendency you use. We still most often use the mean, but it really doesn't matter because they are exactly the same or very close to being the same. If your data or if the distribution is not symmetrical, then it does matter which one you use. And oftentimes, if it's really not symmetrical, the median is a more common value used when your data is not symmetrical. The average doesn't tell the whole story. So for example, here you can see that these distributions have the same midpoint, peak, mid, and average, but they look pretty different. There's a different spread. In addition to a measure of central tendency, we also need to give a measure of how variable the data are. We call this dispersion, how dispersed the values are. Range tells you the difference between the maximum and the minimum, the largest and smallest values. So this gives you the full range of values. One problem with this value, using range for a measure of dispersion, is that it's very sensitive to outliers. So there are cases where it's useful to give the full range of the data, but oftentimes it's better to use a different measure to give your audience or viewer or reader an understanding of the variance in, or another measure of dispersion for your data. Another measure of dispersion is variance. This gives the average of squared deviations. So a deviation is when you're looking at how different each value is from the mean. So you literally just calculate the mean, and then you take each value in the set of data and get the difference between the mean and that value, and that's called a deviation. Since some values will be greater than the mean and some values will be less than the mean, then you have positive and negative values. That's why we square these little deviations. So you take a deviation, which is just the difference between the value and the mean, and then you square them. And then you average this, all those little squared deviations. Now, one problem with the variance on its own is that because you've squared the values, you're now in squared units. So if your original measurement is in centimeters, your variance will now be in squared centimeters. So we then take the square root of the variance, and this is called the standard deviation, to get back into the original unit. So if you were in squared centimeters for the variance, you'd take the square root and get back to centimeters for the standard deviation. The standard deviation is a very common descriptive statistic that gives a useful value of dispersion. Standard deviation is probably the most used value of dispersion when a data set is normally distributed and is nice and symmetrical. If your data is not symmetrical, another concept that could be useful is the interquartile range. And this is the range from the first to the third quartile. Now remember, there's a difference between your full population and a sample, which is just a part of a population. And without getting into the mathematics or the whole reason why, just note that the equation for standard deviation is different based on whether you're calculating the standard deviation of the full population or estimating the standard deviation of a sample. And the main difference is this value on the bottom here. So for the full population, you have the full population, the whole thing. Whereas in the sample standard deviation, the value on the bottom here is the sample number minus 1. And because of this difference, whether your number of individuals is the full population or your sample size minus 1, it makes a difference which equation you actually use. So just make sure when you're using a statistical package, including Excel, that you pay attention to this and that you're using the correct equation, the correct estimate if you're estimating a sample. Most examples you would ever use in biology, you would need the sample standard deviation. And just make sure you use the correct one. What do I mean when I use the word quartile? Well, your quartile is just taking a quarter of your data. So if you have all of your data, you can split it up into 4 parts, and each of those parts is a quartile, or 25% of the data. The first chunk of 25% is the first quartile. The second chunk is the second quartile. The third chunk is the third quartile, and then the last chunk is the fourth quartile. As you can see shown here, the middle quartile, Q2, is right in the middle of the data set, right? So Q2 is also the same thing as the median. So the median is halfway between, right, the lowest and the highest value. So the median is right in the middle. So the median and Q2 are the same thing. The interquartile range is taking the middle 50% of the data. So we want from the first quartile is like the value where 25% of the data is less than that value, right? And then the third quartile is where 25% of the data points are greater than that value. So in between the first and third quartile is those middle two quartiles. And in terms of, you've got 50% of the data. So half of your data fits in the interquartile range between quartiles 1 and 3. So now let's look at an actual sample of humans and their heights. This is a sample from the Department of Physical Education and Psychology from York University, a sample of 112 females and 88 males, and their heights measured in centimeters. And so here's the height on the x-axis, and the frequency is how many individuals had that height. So this is what's called a histogram, and it shows you that distribution of values. So you can see here that both distributions, there's the females in purple and the males in green, both distributions look pretty symmetrical. And if we compare our mean, median, and mode, if it's truly symmetrical, if it's a normal distribution, they should be the same or very close to the same. And you can see that for the males, the values all are identical, and the females, the values are very similar. So assuming that it's a normal distribution and using the mean seems like a fair way to go for this particular distribution. The typical male, using again the mean, is 13 centimeters taller than the typical female. How do we know if that amount of difference is meaningful? Are these truly different or not? So far we've been talking about descriptive statistics. When you use the word descriptive statistics, I'm just talking about useful measures that can describe the sample or a population. So it's purely descriptive. You're just describing what you have. Inferential statistics, on the other hand, are things that we use to make predictions or generalizations based on a sample. If we're using a descriptive statistic to talk about how spread out our data are, and you have a normally distributed data set, you might use something like a standard deviation. If you have non-normal data, you might use an interquartile range to represent how variable your data are. There are some other measures that can be used to talk about how well we did estimating a sample mean that also gets confused with descriptive statistics. So there are some inferential statistics that can help us understand how well we estimated the mean. People often confuse these with descriptive statistics, but the standard error of the mean and the 95% confidence interval are not descriptive statistics. These are inferential statistics that help us understand how well we estimated our mean. So the standard error of the mean, the equation for that is simply the standard deviation divided by the square root of the sample number. What it represents is basically, if you have a normal distribution, the standard error of the mean is 68%, is a 68% confidence interval. And so basically what that's telling you is that there's a pretty good chance that there's a 68% chance that the true mean is within that calculated interval, the actual population mean, not the sample mean. So the sample mean that you calculated, there's a 68% chance that it's in that interval that you calculated. The true mean is also in that interval. It's a very standard measure that's given there are many statisticians who will tell you that a 95% confidence interval is more useful than a standard error. And then some will say it's better to just give the standard deviation to give variation around your mean and use other kinds of statistical tools to tell you whether something is, two populations are different or whatever. And we'll get into that more in the video and inferential statistics. But I wanted to bring up these measures because they're often confused with descriptive statistics. The idea of a confidence interval is actually not the easiest concept to understand quickly, and I really recommend that you take a statistics course to really fully understand it, and it's okay for the purposes of this course if you don't fully understand what it represents. I've also given you an option of a tutorial you can do that is a kind of an interactive tutorial using fish as an example, just on the computer, that can give you a sense of what a confidence interval is and what it represents. But basically, what a confidence interval is where you're taking, if you take a sample of whatever, we could say fish, right? Just like in the tutorial. So you take a sample of fish, let's say we take 10 fish, and then you calculate a mean. And then you take another sample, a different sample, a random sample of 10 fish, and you calculate a mean. And then you do this over and over and over again. A 95% confidence interval is giving you basically that 95% of the time that you calculate this interval, you calculate an interval that the true mean is actually in that confidence interval. Again, it's a pretty complicated concept to really get easily, but basically what we're saying is that with a 95% confidence interval is that there's a 95% chance that the interval we calculated actually has the true mean in it. So it gives you a sense of like how well you did figuring out in the estimate of the mean that we calculated from our sample, how good is that relative to the true mean. A confidence interval is often misunderstood, and people often think that it contains 95% of the data, but that is not what a confidence interval is. This image here shows on the left a small sample size, an n equals 5, so there's just 5 points, and you can see that the 95% confidence interval is pretty wide, and you might misinterpret this to think that, oh, those confidence intervals actually contains 95% of the data. But you can see that when we take a larger sample size, those confidence intervals get smaller. On the far right, you can see that with a sample of 150, the confidence intervals are really quite small. They're actually a little hard to see. And obviously, those intervals aren't containing 95% of the data. So again, remember the confidence interval is telling you how confident we are that we did a good job estimating the mean of the population. So there is a true population mean out there. We don't know what it is. We're trying to estimate the mean of the population by taking a sample. So our sample mean is not always going to be identical to the population mean. And the 95% confidence interval is just telling us how good a job we did at estimating the sample mean. And so when the confidence intervals are smaller, that's a good thing. It means we did a better job at estimating the mean. When you have a larger sample size, you have a higher chance of actually getting closer to the true mean because you're capturing more of the possible variation out there. With a small sample, you could just randomly pick a lot of big samples or a lot of small ones, or there could be some other weird thing that happened because with a small sample size, it's possible to not get a perfectly representative sample. So our confidence interval is wider. We're less confident in our estimate of the population mean.